All right. Welcome to Chapter 5 of Heron and Freeman. I narrated for this course at Florida International University. Evolution is ultimately the study of diversity. It's a core part of biology that links variation within species to broad-scale patterns of variation, microevolution to macroevolution, and population genetic processes on to large-scale changes over geological time. This section of the course, it's a bit of a transition from where we were before the first exam, looks at variation within populations and how it can change over time as a function of changes in allele frequency. As biologists, we're fundamentally, I think, intrigued by variation, and as humans, we may be particularly intrigued by that variation among us. Basketball shows just how variable people can be. But if we compare humans to other animal species, we actually find that we're less variable than many other organisms. If we use the coefficient of variation, which accounts for the amount of variation after normalizing the mean, we see that many other animal species are actually quite a bit more variable than we are. And variation in the biological world is ubiquitous, from flowers that vary in color within a species, to different colored sea stars, to snakes that vary in their color and banding patterns. In humans, a lot of genetic variation is tied to disease. The majority of you are interested in careers in medicine and allied health fields where understanding the genetic basis of disease is increasingly key to our capacity to treat it and to understand it and to care for it with empathy. Likely you've learned about one common human disease that's both important in the history of understanding genetic basis of disease and one of the most debilitating genetic diseases we have, sickle cell anemia, which is a disease of red blood cells, a malformation of hemoglobin. In areas that have high endemic rates of malaria, sickle cell anemia actually confers a benefit, some genetic resistance to malarial infection that seems to be an important part of the dynamics of this disease. So this is a molecular image of hemoglobin. But before we delve too deeply into any particular disease, I'm narrating this chapter in part because I want you to have a little bit of review of genetics. In the big picture, we started this course in part with Darwin. We worked through an example of HIV. It was 100 years after Darwin, roughly 100 years after Darwin discovered evolution by natural selection as a process, that we actually understood the genetic basis of heredity on a molecular basis as DNA. DNA is a molecule that carries our genetic information. It's a double helix, and it comes in deoxyribonucleic acids of four flavors, A, C, G, and T. And these four letters can form words that code for proteins. We'll delve into this as review in a few minutes later in this. But this double helix nature of DNA allows it to be replicated with high fidelity, making it the ideal genetic material and explaining the particulate and segregating nature of inheritance that Mendel, unknown to Darwin, at the same time, about a century before the discovery of DNA, had described in his two laws of inheritance that we covered in class last week. So DNA is the molecular material, and it's formed into chromosomes, which we often depict like this. Chromosomes are long, hundreds of millions to billions of base pairs of DNA. If we stretch them end to end, they would stretch for meters, and they have to be packaged into condensed molecules so that they don't flop around and take up all of the space in our cells. So we frequently depict them this way. They are linear molecules, even if they're condensed around histones and other packaging proteins. And because they're, particularly in eukaryotes, linear molecules, they have a characteristic that means there's linkage. There's a physical distance between different genes that code for proteins on the same chromosome. So this is a map of the approximate location of where different genes are on a human chromosome, one of which you've also studied in your genetics labs, the PTC taste receptors. Taste receptors are something that in humans vary substantially. After immune receptors, taste receptors are the second most diverse class of genes in humans. And what one thing tastes like to one person tastes quite different to another, such as cilantro. I happen to think it's delicious. My significant other thinks it tastes like soap. This is not unusual. We differ genetically, and that's a good thing. PTC is well studied. Although there's not PTC in our food, likely it confers the capacity to taste the bitterness in different kinds of mustard plants like broccoli or kale, plants in the mustard family. And how bitter they taste to us is likely linked to different alleles of this gene. And depending on what genotype you have, you may be able to taste the bitterness or not. All right, so PTC, it's a taste receptor coded by the PAV allele or the AVI allele here. Ultimately, we have two copies, one from each of our parents. And this happens because DNA is replicated in most of our cells by mitosis, but in meiosis it creates our egg and our sperm, or egg or sperm in animals, in something like a plant that happens to be a hermaphrodite. It'll be both pollen and eggs. These haploid, only one copy of the genome cells, the egg and the sperm, combine, hopefully not with genetic material of the same individual in a hermaphrodite like plants. In humans, where we are male or female, it takes an egg and a sperm. Through fertilization, you recover a diploid organism that becomes a zygote, and if surviving, will grow into a diploid adult. And if one chooses, go on to do it again. So meiosis is important. It takes the two copies one has of each chromosome from your parents, and your reproductive cells will only get, your gametes will only get one chromosome from one or the other of your parents. So there's a lot of shuffling of genetic material that happens during meiosis. This creates some variability. Later in the course, we'll get into some of the evolutionary explanations for why sexual reproduction is likely favored. Here, we show how the different possible gametes from two mating individuals can recombine to generate different genotypes at this PTC receptor locus. So we can get different combinations depending on which of the grandparents' alleles are passed forward. All right, so this is one, we can look at this as a Punnett square, a way of diagramming, and it's named after George Punnett, a British geneticist who played a role in rediscovering Mendel's work. This is a way of diagramming how particular mating will go forward, and we're going to come back to several of these later in the next couple of weeks. All right, I'm going to shift gears a little bit. This organism, this almost freaky looking thing, is Daphnia pulex, water flea. They're a fascinating organism that can do all sorts of things. They can survive a nuclear explosion in the muck. They can go into a dormant stage and survive for decades in the muck. They are prolific reproducers. They switch between reproducing asexually, essentially clonal reproduction, and then they can reproduce sexually when they run out of resources or they're otherwise stressed. They also happen to essentially be fish food, but they will generate different sorts of intradenticles to help protect them from fish when they smell different pheromone uh, or other things in the water that indicate fish are there and feeding. And as soon as they sense this fish, um, essentially fish spit, as soon as they sense it, a number of genes are expressed to induce the expression of these protective structures on their back to keep them from being quite so tasty to fish. All right, so that's an important form of variation. And the reason to look at this in water fleas, I'm going to go back a couple of slides, is that this is something that's genetically controlled. There's expression of a couple of genes. There's variation in water fleas and Daphnia in these genes, and you can compare populations of Daphnia. This is going farther than the textbook. In some populations, in areas where there simply aren't fish, they accumulate mutations in some of these genes, and they don't upregulate expression quite as strongly when they smell fish. And this is because they've lost the capacity to do that. Populations that consistently have high levels of fish may never downregulate the expression of these. So we can find segregating variation among water fleas. We also have a trait that's plastic. This is something that water fleas only express, if they can express it, in the presence of fish. So before the first exam, I described genetic variation as being something we can partition. The phenotypic variation that we observe equals the environmental variation, or the genetic variation due to the environment, due to the genotype, and due to the interaction of the genotype and the environment. And it's important to keep this in mind. For more complicated traits, likely they're caused by more than one locus, and they have an environmental component. Many of the most complicated human diseases, like diabetes, are somewhat similar to this. All right. Another example that your book walks through is toadflax. This is a fascinating flower. This image doesn't do it justice. It really is a beautiful white flower that in a temperate forest, it's a small little herb, can make a botany student's heart delight. I happen to think it's a beautiful plant. It normally has a bilateral flower. That means you could draw a line through it and cut it in half, with each side being equal. But we can find mutations that make it radially symmetrical. So you can divide it in half, but there's not just one axis through which you can divide it in half. So it's a radially symmetrical flower rather than a bilaterally symmetrical one. And there's a single mutation in a transcription factor that will cause this. Another form of plasticity is the gender development of some reptiles and amphibians, where depending on the temperature they develop as males or females. Interestingly, depending on the population, how sensitive they are to temperature varies. So this graph over on the right is something that we call a genotype reaction norm. Each of these lines is a genotype reared under a couple different conditions. In some lizards where you can rear them parthenogenically, they're actually genetic clones. You can do this in some plants as well. If you take a banana and you make cuttings of it, it's how we propagate most commercial bananas. You can take cuttings of it, and you've got the same genetic individual. They're just cuttings of the same banana tree. And then you can, well, they're not technically a tree, but of the same banana stem, and you grow those different cuttings under different conditions. Or if you're working with a sexually reproducing animal, you take siblings. You can take half sibs and full sibs. This is widely done, say, with cattle. And you take each genotype, each sort of family, if you will, and you grow them under two different temperature regimes. In this case, you can look to see what the percentage of males and females is. And what you notice is that these reaction norms differ substantially. What this means is there's genetic variation in how plastic they are to temperature. This happens to be a fascinating subject to me. This was in part the topic of my own doctoral dissertation. All right. And even if I've used animal and plant examples following the textbook, this is something that likely happens in humans. An example the book gives is in susceptibility to depressive episodes, which is clearly linked to neurotransmitter sensitivity and sort of coded by receptors for neurotransmitters. And people with certain genotypes are more prone to depressive episodes. And it's clear if we just colloquially, we notice that there's variation in how susceptible individuals are to things like depression. Similarly, in perhaps the most troubling disease that we'll face in the US in coming decades, diabetes is very similar. We vary in how susceptible we are to the hazards of the high sugar, high fat, low physical activity Western diet. All right. Another example, this is back to an insect one, is caterpillars, where some brilliant work showed that you can select on how plastic they are in their heat response by selecting for high plasticity and low plasticity lines in their heat shock responses. All right, so there's sort of a jump here. The book takes this as different sections. Ultimately, this variation in plasticity is due to genetic differences. And we're sort of going to structure looking at this for you. So let's briefly go back to DNA. It's a deoxyribonucleic acid. It's got a sugar phosphate backbone, 4 letters. This is what it looks like as a chemical structure. The important thing about it is that A and T have these 2 hydrogen bonds. They bond to each other as different chains. G and C have 3. That makes them slightly more stable. There are high temperature from places like Yellowstone, where there are natural thermal springs. You find organisms that have an overabundance of G and T in their genome because these extra hydrogen bonds make them slightly more stable. And there may be a little bit of selection on genetic content for this reason. All right, so the double helix can be opened up to copy the genetic material. This can be done straightforward during mitosis, or it can be done, similar process happens in meiosis. It's worth noting that in the reverse direction, in one direction you've got a direct copy being made, and in the other it's filled in with the formation of Okazaki fragments. There are some mutations that happen in this. It's generally a high fidelity process, but mutations happen. And this is the basis of the genetic variation that we subsequently observe. One of the most common is T being converted to C, or C to T, where C is converted into something that looks like U, the RNA molecule U, and then converted to T. There are other kinds of changes that can happen. So there can be deamination, the C to T. There can be insertions or deletions that happen during replication. And for them to go forward, you see one strand gets this, one daughter copy gets this change, the other does not. So that's ultimately what a mutation is. And things like UV light, carcinogens, mitogens in the environment, or simply bad luck, all of them can cause mutations to occur. And so this is what an insertion might look like. Now, our genome is filled with a number of copy editors. DNA polymerase itself has a copy editing function, and there are other proteins that will go through our chromosomes and correct mutations as they occur. And the very packaging of DNA onto histones, as well as functions that acetylate and methylate DNA, protect it from a variety of mutations. But if those proofreading and protective mechanisms are deleted, you get elevated rates of mutation. And if we take mice and we make them deficient or doubly deficient in these repair mechanisms, we greatly reduce their survival and give them very, very short lifespans. All right, so I mentioned earlier that DNA is letters and they form words. This boils down to something that I'm going to call the central dogma of molecular biology. The DNA is transcribed into mRNA and then translated into protein. We know that there are exceptions to this. There are retroviruses like HIV that go from mRNA back into DNA. There are a few protein diseases like Kuru or mad cow that can interfere with DNA forms. And there are things like small RNAs that behave in a slightly different way and may modulate mRNAs or proteins. Biology is full of complications. But the general way that we look at genes is as being open reading frames, DNA that can be transcribed into an mRNA and turned into a protein. DNA is read in words of 3 letters, and this is the code. So there are 64 possible combinations of the 4 letters. There are only about 20 amino acids and 3 stop codons. I said before that the start is always methionine in all eukaryotes at least, and I believe it's true in all bacteria, although you're welcome to dig deep and find an exception for me. In all eukaryotes, the start of an open reading frame is a methionine. And usually 25 bases before it, there's something called a TATA box, a TATA, that RNA polymerase binds to as it's basically a naked promoter. And it allows RNA polymerase to find this AUG, this start. Any of these 3 stops will lead to the termination of RNA polymerase. It will fall off of the open reading frame, and that will terminate the growth of the RNA chain and lead to a shortened amino acid chain. Now, in many eukaryotes, there's the complication of something called introns. These are gaps that are read into the first RNA molecule, but then cut out as it becomes mRNA. They're excised so that only the exons are actually converted into amino acids. So you can get mutations in the introns that are silent. They have no function, or they have very little function. They may affect whether or not an intron is spliced, because in some proteins there's alternative splicing possibilities where different exons or introns are used. The other thing worth noting is that this is a degenerate code. You see that there are multiple ways to get a leucine, an isoleucine, a serine, etc. This means that mutations particularly in the 3rd base pair, and in a few cases in the 2nd base pair, will cause no change in the amino acid. These mutations are silent, and these are one of the most useful things we have in population genetics, because we can compare these silent mutations in the 3rd base pair or in introns to the mutations we observe in that cause an amino acid difference. These are non-synonymous or amino acid substitution changes. So we can compare non-synonymous to synonymous or silent substitutions to look at what the significance of mutations are. All right, so I called this the central dogma of molecular biology: DNA to RNA to protein. This is what a growing chain will look like. So this is how it's transcribed. So this is another look at the code. I am not going to expect you to memorize different parts of this. You may or may not need to for biochem. It's not required here. But it's important to understand the significance of it as a degenerate code. All right, so some of these changes between the 4 letters of DNA are more likely to happen than others. In general, changes within a class from one purine to another or from one pyrimidine to another are more likely to happen than the others. So transitions are more likely to happen than transversions. So we can use the difference in the rate at which these happen, or the likelihood that these happen, to develop statistical models of how likely a particular DNA change is to occur. We can also add in whether or not that change is a silent mutation or not. All right, so that's what single base pair changes might look like. Other types of changes do happen quite frequently. So one thing that can happen during meiosis is something called crossing over, where these 2 strands, they're long, floppy pieces of DNA, particularly when they're being opened up or taken off of their histones to be copied. And sometimes they cross over, and sometimes they do this in an unequal fashion. And that can create duplicate copies where one daughter lineage gets none and the other gets 2. So this creates immediately a new gene. It's very similar to the existing gene, and if its expression or its function depends on dosage, the duplicate may be a problem. But if it's relatively insensitive to dosage, it's possible that it will accumulate mutations, that it'll be knocked out and become a pseudogene, or it may evolve a new function, or the 2 copies may subdivide an ancestral function, particularly if a protein does multiple things, with one version of, say, hemoglobin being expressed in general cells and another being expressed in, say, developing fetuses, where it's essential that they draw oxygen more strongly so that they can take oxygen out of the mother's blood. So having 2 copies of one ancestral gene can allow improved function. And these are some of the most important mutations that we have. We can also get new genes by retroposition, where that mRNA is then, for one reason or another, due to viral activity or just a mistake, reintegrated into the genome. And it might be integrated in the same spot, or just as likely, if not more, into a new spot. Now, these behave quite differently than tandem duplications. For one, they're not necessarily in the same spot in the genome. So if I give you information, particularly on an exam question, that says they're not in the same place, that's a key that it's a retrotransposition. The other thing that happens is these introns are spliced out as part of the process. So if I tell you that 2 genes are the same in their exons, but different in their introns, it strongly suggests that a retrotransposition occurred because those introns are lost as part of that. So this can give us important information on how duplicates arose. So in something like the Hox cluster, where you've got several genes next to one another, the fact that they're together suggests tandem duplication, as well as the fact that they're under strong selection for their order on the chromosome affecting their function. All right. I happen to think dogs are fascinating. I can't help but use the book's example here. There are a number of dog breeds like corgis and other dogs that have short legs, and this has a genetic basis, the FGF4 retrogene. So short-legged dog breeds tend to have DD genotypes at the DD allele at this locus. And this is actually a good reminder. You should review the difference between an allele, locus, and a genotype. I made that point in class, but this is another good time to check in on that. Another kind of mutation is due to an inversion, where the whole chromosome breaks and reanneals, but reanneals in the opposite direction from how it originally occurred. So you've got this hairpin forming, and then it breaks and is reinserted. So these genes are now in the opposite order compared to the others. Now, if you have an individual with this new order try to mate with an individual in this order, it creates problems during meiosis. When the two copies of the chromosomes line up during meiosis, they no longer match one another very well. So this can actually cause reproductive problems during sexual reproduction in the next generation. So individuals with this inversion are often going to have higher fitness if they only mate with individuals with the same inversion. And this is likely one factor that's important in speciation. In fact, two of the wild species of chickpea that I study, both from southeastern Turkey but different parts of it, we think are incompatible with one another in part due to an inversion that separates them. When we cross them to one another in our breeding program, we find that some won't give viable offspring, and we think it's due to an inversion like this that screws things up during meiosis. Fruit flies happen to have some fascinating inversions. They have a number of them, and we've noticed that fruit flies from Europe, South America, North America, and it's not described in the book, but it also happens in Australia, all have similar patterns of inversion, where populations from more equatorial regions tend to have similar frequencies of inversions across the world, and at higher latitudes, a different frequency of inversions. And there seems to be selection on this. And it's likely not the inversion itself that's being selected, but genes being, alleles of genes being carried to high frequency within these inversion groups. And the inversions help to keep flies from different latitudes mating with one another. Now, the book, and I have to cover this given my passion for plants. In plants, another process of mutation is quite common. It's rare in animals, but it does happen, particularly in hermaphrodites like earthworms. This is polyploidy, and even in the lineage leading to humans, in vertebrates, it likely happened. That's likely how we have 4 copies of the Hox genes, due to a very ancient, before the colonization of land, tetraploid formation in fish, what was then an organism that looked like fish and gave rise to tetrapods. So the way polyploidy works is you've got a 2N genome. You end up getting non-reduced there are actually a couple mechanisms by which it occurs, but the most common is that you get non-reduced gametes, 2N gametes that are supposed to be 1N. Instead, they form a 4N offspring, and that 4N offspring then has 2N haploid gametes. So you've got a doubling of the amount of DNA. Now, if a 2N gamete tries to mate with a 1N gamete, you end up with a triploid. And in general, triploids are sterile. Rarely they may occur. So what happens is, if you've got a lone plant that's unable to reproduce because there are no other plants around, or an absence of bees or wind to pollinate them, the at a rate of maybe 1 in 1,000 or 1 in 10,000, gametes, and this actually probably happens in our gametes too, gametes form that are 2N. It's simply an error in meiosis, just a mistake that happens where these 2N gametes form. If they reproduce with one another, you initially get these 4N individuals. Because triploids, 2N, 1, and 1N gamete pairings, or triploid zygotes, are generally infertile or of much lower fertility, you would very quickly separate 4N from 2N populations. So you can get immediate species formation because the new tetraploid individuals are of low compatibility with those of the ancestral diploid population. So there are a number of plant species where we've observed both occurring with one another. And this is very common, if not ubiquitous, in plants. Among our crop species, corn or maize, soybean, those are the two most widely grown plants in North America, are both tetraploids. Most of the wheat that we eat for bread or for flour, it's usually a hexaploid, 6 copies, although sometimes there are forms of wheat that are only 4 copies as well, and even some ancient wheats that are 2 copies. Peanuts, cotton are also both tetraploids. Potatoes are tetraploids. And there are a number of other crops. Sugarcane, an important crop here in Florida and across much of the Caribbean, is actually a much higher ploidy. There are different sugarcane varieties with much higher 8 and above ploidy. Most strawberries are octoploid as well. So a great many of our crops have this characteristic. It's simply common in plants because of the way they tend to self-fertilize. So there's some seminal work from Justin Ramsey using a plant called yarrow, where he looked at 4x and 6x individuals and their survivorship in different places, showing that in more stressful habitats, these 6x individuals were more had higher survival. All right, so if we step back to look across organisms, we see that there are big variations in how common mutations are, where particularly retroviruses and RNA viruses have extremely high mutation rates. It's because RNA, reverse transcriptase, is a very error-prone way of reproducing, and RNA itself, because it's not double-stranded but single-stranded, is inherently unstable. We see that mutations are ubiquitous, and that I think contributes to a lot of the diversity we observe. I want to make a quick mention of, I've never asked an exam question on it, but one kind of common experiment, it's shown up on the MCAT before, is a mutation accumulation experiment. I happen to like this one from Arabidopsis. A good friend's spouse did this work as a postdoc. You take seeds from an original line and you keep each subsequent seed separate, and you let them accumulate mutations. In the Arabidopsis work, they grew them under non-stressful conditions for 30 generations, and they started to see mutations show up. Another way you can do this is induce mutations, and there's lots of work in fruit flies, in zebrafish, in Xenopus, a kind of frog, in Arabidopsis, in mice, where researchers have mutagenized them to generate variation. With that, I'd like to stop. Enjoy. I'll see you in class.